This is the second video to describe Lab 3 in the Citizen Science program. An assay commonly used in clinical laboratories to test the potency of antibiotics and drugs is a filter paper disc agar diffusion procedure, commonly known as the Kirby-Bauer test. A filter paper disc agar diffusion method is also used for determining the potency of antiseptics. In this exercise, you will have an opportunity to determine both antiseptic and antibiotic potency with a modified Kirby-Bauer test. Discs of filter paper saturated with antibiotic solutions in the same range of concentrations achieved in the human body are placed on an agar plate heavily seeded with the test bacteria. The antibiotic on the disc diffuses through the agar to form a concentration gradient. When incubated, the bacteria grow in a smooth lawn of confluent growth until they reach an inhibitory concentration of antibiotic. This creates a clear zone around the antibiotic disc, called the zone of inhibition. Our experiment with an overnight incubation will allow us to determine the general sensitivity to the antibiotics and antiseptics being tested. Here in this image, we're just seeing stacks of Kirby-Bauer assays. These are not our plates, but I just wanted to point out right here that these right here, these are the filter discs that we just heard about, okay? And remember, this is the clearance. We talked about that already, okay? This is the lawn. All the fuzzy stuff is the lawn, okay? And the clearance is the part where the microbes could not grow because whatever was on the filter paper inhibited their growth. Label the agar side of your plate on the outer rim of the plate, indicating your section number, student number, and date. In addition, place a small orientation mark at the top of the plate to be used later to orient the plate with the template. Using a sterile stick, take a colony from your isolate plate and mix with 250 microliters of LB broth in a microcentrifuge tube. Pipette 100 microliters of the culture into the middle of the agar, your plate labeled isolate. Shake about 5 to 10 glass beads onto the plate and replace the lid of the Petri dish in a gentle random motion. As evenly as possible, let the beads spread the culture across the agar. Once you have prepared your plate with the bacterial cultures, proceed to the antibiotic station. Place your plate on the template provided with the orientation mark aligned. This will give you a guide for placing the appropriate impregnated discs in the corresponding positions. Here is the here's the code for what substances we are going to use here to test antibiotic and antiseptic resistance. So take a look at the word roots. The first 4 words are in one group. Do you think those are the antibiotics or the antiseptics? Then there's 5. What is that? And then there's 6 through 9. Think about what you think those are. Are those the antiseptics or the antibiotics? Using the sterile forceps provided, immerse one of the filter paper discs into the antibiotic solution of kanamycin located in position 1. Blot the disc thoroughly on a piece of clean towel and place it in the area labeled 1 on the agar. Be careful not to touch the forceps to the agar of the plate. Repeat steps 10 and 11 for the remaining 8 antibiotics and antiseptics. Invert the dish and incubate. We will observe and discuss the zones of inhibition in the next lab session. When a bacteria is isolated from the environment or from an infected individual, it is important to identify the specific type of bacteria that is being dealt with. One of the first steps to identifying bacteria is to perform a gram stain. Most bacteria can be classified into one of two groups, each of which will react differently to gram staining. Gram-positive cells appear purple and gram-negative cells appear pink due to the difference in the outer layers of the bacterial cell. This differential staining also allows the shape and arrangement of the bacteria to be better visualized. So thinking back to the very first video with the handprint with the crazy microbes growing all over it. When we are thinking about how bacteria look, we can look at their membrane structure as one measure of what's called their phenotype. So the word root type means type, and pheno means physical. So the physical type, how does it appear? Okay. The thing to notice as you gaze upon this image is not the presence of the outer membrane, but rather the thickness of the peptidoglycan layer. Okay, so notice how in gram-positive that layer is brown and it's thicker and it's on the outside. And in the gram-negative, it's narrower. It doesn't really matter that it's inside. What we want you to understand is that materials have to diffuse through membranes, and if the membrane is thicker, that's harder, okay? So we are going to use this as one of our tools for identification. Through the staining process I will describe in just a moment, we'll be able to find out whether or not the bacteria are gram-positive or gram-negative. If they're gram-positive, then we have a darker appearance, the purple, and if they're gram-negative, then less dye is taken up and what we see is pink. But what you notice here also is that we can better determine the shape of individual bacteria, and here we have two types. We have bacilli and cocci. So the bacilli or bacilli are the rod-shaped bacteria, and the cocci are the circular ones. The easiest way to think of that is C stands for circle, and we will have diagrams of all the possible shapes. So those shapes, in addition to the presence of or in addition to the identity as gram-negative or gram-positive, are two tools we're going to use. Using a sterile stick, take a colony from your environmental isolate plate and mix with 25 microliters of LB broth in the microcentrifuge tube provided. Remember, the microcentrifuge tube is that little tiny one with the flip cap. Label 3 microscope slides, E. coli, M. luteus, and isolate. Using the greased pencil, draw a circle on the slide to create a well. Pipette 5 microliters of each liquid bacterial culture into this well on the appropriately labeled clean microscope slide. Spread the drop into a thin film using the pipette tip and allow to air dry. This can take several minutes and will result in a white film on the slide. When the smear is dry, pass the slide through the flame of a Bunsen burner 3 times to heat fix the bacterial cells to the slide. The teaching fellow will have the burners set up for you. When the smear is dry, pass the slide through the frame. After the slide cools, cover the smear with a thin layer of crystal violet solution and allow it to sit for 1 minute. This binds to that peptidoglycan. Rinse the slide with water and then shake off the excess. Add a few drops of iodine to the solution and allow it to sit for 1 minute. Rinse the slide with water and then shake off excess water. Add a few drops of the 95% ethanol. Tilt slide back and forth between draining off the ethanol. Repeat step 11 until the ethanol runs clear. Rinse the slide with water and then shake off the excess. Add a few drops of safranin and allow to sit for 1 minute. The pink binds to the cell membranes. Rinse the slide with water and then shake off excess. Dry by carefully blotting between pieces of paper towel. Pass the slide once through the flame and examine the slide with the microscope. The teaching fellow will have several microscopes set up to assist you.